When deciding which types of data to include in your presentation, there are a few important things that you should keep in mind. One of the most important is that figures and graphs are more effective for displaying results than text. However, you have to make sure that the figures that you use are simple, that they're easy to understand, and that both your slides and your figures are uncluttered. Whenever you use a graph, you also have to remember to explain each of the axes and the variables that are shown in the graph. And you must always include a reference for all figures that you use, whether these are from papers or websites. If a figure is not your own, or if you made a graph or a figure using data from another source, then you need to cite that source. This first figure is from a paper, and as you can see, I've included a short version of a reference that does not include the title of the paper, and it only has the last name of the first author. As you can see, these references can be in small font size. This is 10-point font, and you can include it at the bottom of the slide because you don't want it to distract from the rest of the content of the slide. In addition to including a reference, you should include titles that reflect what is shown in the figure. You cannot simply write something like data or results as a title. That's not considered professional. You also need to include any information that will help the audience to follow along as you describe the figure. For example, this figure shows mouse embryos that were stained with neurofilament, which is a neuronal precursor marker, in green, and with the markers SOX10 and MITF in blue and red, which label neurocrest cells and also pigment cell or melanocyte precursors. As you can see, a label is included in the figure that matches the colors that are used in the staining. In addition to that, I also indicated that the abbreviation E stands for embryonic day. Here's another example of a figure, but this is from Wikimedia Commons, not from a paper. But even though this figure is not from a paper, I've also included a URL for the website where that figure is found, and you need to make sure that you always do that. I also want to take this opportunity to caution you to be careful where you get your figures. You need to make sure that the figures that you're using have not been posted illegally and that they are safe and free to use. In addition to using figures that show data or structures, figures can also be useful to summarize processes or main findings, and you can either create your own figures for this purpose or use a figure from the web like the one that is shown here. As you can see, this figure is very simple and even unsophisticated, but it is very effective for explaining the basics of the process of telomere shortening. You can also make your own figures to explain concepts. For example, let's say that you wanted to explain the difference between additive and synergistic genetic interactions. Rather than reading definitions of these terms from a slide, it would be more effective to explain these with a figure. For example, for genes that exhibit additive genetic interactions, the phenotype of the double mutants would be what you would expect from the addition or the sum of the phenotypes of the single mutants. Whereas for genes that interact synergistically, the phenotype of the double mutant would be more severe than you would expect from the addition of the phenotypes of the single mutants. As you can see, explaining these with a figure not only makes it easier for the audience to visualize what these concepts mean, but it also makes the presentation more interesting.